Hello student, good afternoon. Today we will start the new cell organelle, that is Golgi complex. Golgi complex or Golgi complex. This is the cell organelle. In the eukaryotic cells, 5 or 6 cell organelles are present. Golgi complex, endoplasmic reticulum, ribosomes, lysosomes, mitochondria, all these. One of that is the Golgi complex. Golgi. Golgi in 1891 first of all recognized it in the nerve cells. This Golgi have described the 1891 on this name, that is the Golgi complex. He has detailed study of Golgi complex. First of all, recognize it in the nerve cells. First of recognize in the nerve cells of the brain, owl, and the cat. OWL, owl, goat, and the cat. After the name of its discoverers, Golgi complex was variously named of Golgiosome, variously named of the Golgiosome, Golgi bodies, and Golgi material, Golgi membrane. All this nomenclature of Golgi complex, that is Golgiosomes, Golgi bodies, Golgi material, and Golgi membrane. The Golgi complex of the cells of plants and lower invertebrates is usually referred as Golgi or Golgi body or dictyosome Biosynthesis of polysaccharides, packaging of cellular synthetic products, proteins, that is proteins, production of exocytotic vesicles, and differentiation of cellular membranes. There occurs a complex organelle called Golgi apparatus or Golgi complex in the cytoplasm of animal and plant cells. Both the animal cell as well as plant cell also, this cell organelle. Only eukaryotic type of cells. In prokaryotic cells, these cell organelles are absent. Only ribosomes are present. The dimensional view of Golgi complex. Tubules, membrane of Golgi complex, secretory vesicles, and cisternae. The Golgi complex found in all cells. Except the prokaryotic cells. Because example, bacteria, mycoplasma, and blue-green algae. In this organism, these prokaryotic cells are present. Blue-green algae. And eukaryotic cells of certain fungi, sperm cells of bryophytes and pteridophytes, cells of the mature shoots, tubes of plants, and mature sperms and red blood cells of animals. In animal cells, there usually occurs single Golgi complex. However, developing oocytes of the chordates may contain many Golgi bodies. In animal cells, The Golgi complex is a localized organelle. Example in the cells of ectodermal origin. Cells of ectodermal origin. The Golgi complex remains polar and occurs in between the nucleus and the periphery, and in the nerve cells it occupies a circumnuclear position. Structure of Golgi bodies is the morphology, is the structure of morphologically each type of organelle of Golgi bodies is disc-shaped and consists of central flattened plate like compartments or cisternae, peripheral network of the interconnecting tubules and peripherally occurring vesicles and Golgian vacuoles. Cisternae, tubules and vesicles. That is the combination of this made for Golgi complex, body of Golgi complex. Number 1, cisternae. A cisternae is a sac or cavity filled with fluid contents within a cell or organism. A Golgi bodies or Golgi complex usually consist of 4 to 7 or 3 or 12 flat tubular or filamentous cisternae, which are closely held in parallel bundle or stacked one above the other. The membrane of cisternae per Golgi body may exceed up to 30 or more in lower organisms. All the cisternae of a stock of Golgi bodies or Golgi complex are slightly curved so as to give a concave and a convex face of the whole stalk. Each cisternae of stalk is enclosed by the usual 3 cisternae are separated from each other. The intercisternal space of about 100 to 150 angstrom unit. Width. Each cisterna consists of a central plate like region saculi of a 0.5 to 1 micron diameter, which is the fenestrated at its peripheral margins due to its extension into network of tubules. The cisternae have some definite polarity. The secretory vesicles are formed from the distal poles of cisternae. Second is tubules. From the peripheral area of the cisternae arises a complex anastomosing flat network of tubules of 300 to 500 angstrom unit diameter. Cloches and Juniper scientists have described 1969 have compared this tubular network to a disc-less. Third is vesicles. The vesicles are smooth droplet-like sacs which remain attached to tubules at the periphery of the cisternae. The are of following two types: smooth vesicles and coated vesicles. Smooth vesicles and coated vesicles. Smooth vesicles are of 20 to 80 millimicron diameter. They contain secretory material, secretory vesicles, and are budded at from ends of cisternal tubules. Within the net, often more than one tubules connect to the and presumably fill a single forming vesicle. The coated vesicles are spherical protuberances about 50 millimicron in diameter, and with a rough surface. They are found at the periphery of the organelles, usually at the ends of singly tubules, and are morphologically quite distinct from the secretory vesicles. Golgian vacuoles. The Golgian vacuoles are large, spacious, rounded, sac-like structures occurring at the distal ends of cisternae. They are produced by vesiculation of saccules of the cisternae. This is the structure of the Golgi body or Golgi complex. Okay, thank you.